Good morning everybody. Welcome to class 9th. As in the previous class, we have discussed about the chapter, the cell, its structure and functions. And in 9th class, we have the basic chapter, that is the fundamental unit of life. We know the basic structural, functional and fundamental unit of life is cell. It is called the functional unit because all the important functions of the body like excretion, digestion are done by a cell. And it is called the structural unit because it is the building block of life. It makes the structure of an organism. Now the cells, they remain undiscovered for a long time because they are microscopic in size. Cells, they can be divided into two categories. On the basis of the number of cells present in an organism, there are two kinds of organisms. One is unicellular, another is multicellular. As the name suggests, uni means single. So the organisms which are formed of one cell, like Chlamydomonas, amoeba, they are formed of one cell. Multicellular, multi means many. So here the organism, it is made up of many cells. A very good example is of a human being. We know in our body we have various kinds of cells like nerve cells, fat cells, egg cells, muscle cells, etc. Now, before we move further, there is one experiment which is in your practical files also, and we will be doing that in the class. That is how to make the temporary mount of onion peel. Now, what do you mean by temporary mount? Temporary, there are actually two kinds of mounting. One is permanent. Permanent slides we have in our labs which we are using years after years. But here we are concerned with the temporary mounting of slide. That is, we'll be making the slides in practical period and we'll be showing it to the teacher and we can wash it. Now for this chapter, here we have to do we have to make a temporary slide of onion peel. I'll be going to upload one video also which will be very clear to you how to make that. So what is onion peel? Like I've taken onion here. We can take the peel out of it. We can remove the part from the inner side of the hair. This one is the peel. You can see this peel. We will put this peel in water and it will be dipped in water for some time. After that, we will be putting a stain called safranin for it. Stain is which will provide color to the cell because otherwise cells are colorless. So we have to provide them color. So we will put safranin stain in it. After that, we will be holding that peel and we will keep it onto the slide and we will put glycerin. That glycerin is colorless and it is hygroscopic. That means it has water content in it. And we will be covering it with a small square like thing that is called cover slip. And we will observe this slide under a microscope. What we are going to see, we will be able to see small cells, small rectangular cells like this, as they are referred to as tiny compartments by Robert Hooke. We know about the discovery of the cell.
Go.
So here we see these rectangular cells having the nucleus in them. So this we are going to see in the labs, that onion contains such kind of cells. So with this we know the discovery of the cell was made by Robert Hooke. And in which year? You remember, in the year 1665. And what was the material he has taken? He has taken the thin slice of cork and he observed it under his self-made microscope, and he saw little compartments to which he named it as cell.
Go.
Now the next topic is, what is the structural organization of a cell? That means that what makes the structure of an organism. So here the lesson will be divided under 3 topics. One is plasma membrane. We know we have done this in 8th, the outer membrane of the cell of an animal cell. Whereas in case of plants, there is an outside covering which is known as cell wall. So we will be covering cell wall under this topic only, under the membranes. Now the second thing is very important part, which is the control center of the cell, that is the nucleus, the master organelle of the cell. And the third thing is the cytoplasm. The cytoplasm, we know it is the fluid part of the cell which has the suspended cell organelles in it. And various cell organelles we know, like mitochondria, ER, Golgi body, lysosomes, etc. And in this class, we'll be going to we'll see those in details, their structure and their functions.
Go.
Now starting with the very first one, that is the plasma membrane. It is also known as plasma lemma. Now what is the importance of this plasma membrane? This plasma membrane, it separates the inner contents of the cell from the outside environment. So that is the first function. Separates the inner content from environment. And the second is, it controls and it permits only the exit and entry of certain substances. It permits the entry and exit of some materials through it. And those some materials, what can be those? It can be carbon dioxide, oxygen, water, etc. It's called selectively permeable membrane.